There we go. Question number 3. The diagram shows a plant cell with some structures labeled. Which structure is the cell vacuole? So it's given us this. There's a lot of letters here, so it might be confusing. So P is not the vacuole. It's pretty clear it's not a vacuole. R is also not the vacuole. S, nope. T, yeah. So the vacuole is just this going to be very large, much larger than any of the other organelles, and it's going to have this kind of shape, this kind of like, I wouldn't say blocky, but kind of long and wide. It takes up a lot of space, that kind of shape, and this is the vacuole. If you have things like this, it's going to be mitochondria and chloroplast. This is, these are chloroplasts. This is the mitochondria. You can see this is chloroplast because of the lamellae and the thylakoids, and then this is going to be the mitochondria because you can see these invaginations, the cristae. But yeah, it should be pretty clear that the vacuole is going to be much bigger than the rest of them, and yeah, pretty easy option. So D. Okay, which structure is the site of photosynthesis? Well, we know the site of photosynthesis is going to be the chloroplast, and we just have to pick the one that is the chloroplast. And as I mentioned before, P, this one, P is actually pointing at the thylakoid, which is the true site of photosynthesis, where the chlorophyll is located and the photosystems are located. But in general, P is pointing at the chloroplast, so it does mean that P would be our answer, where photosynthesis occurs. There we go. Okay, triple I. Which structure is the cell wall? Well, that should be pretty easy for us to see. It's S. If you're wondering how I got S, you're just looking at this thing right here, the outside cell wall. I'm sure you can figure that out yourself. Okay. Next one, structure U is the site of protein synthesis in the cell. What's the name of structure U? Well, protein synthesis is associated completely with ribosomes, so we should know that it is ribosomes. We know that chloroplast is for photosynthesis, mitochondria for respiration. Starch granule has no relation to protein synthesis. Okay, B. Cell is shaped like a cube. Each side has a length of 0.053 millimeters. Calculate the surface area to volume ratio of this cell. Interesting. So it's basically asking us to calculate the volume and surface area respectively, and then you have to put them into a ratio. And this is pretty easy to do now that it's mentioned that the cell is shaped like a cube. So first thing you have to do is give the volume. So it's a cube and we have a side length, so it's pretty easy to get the volume. We're just doing the side length, which is 0.053 times 0.053 times 0.053, or 0.053 cubed. You have your calculator, you can do this. It should get you an answer of 0.000148877, which could be rounded to 0.00015, but I wouldn't recommend rounding anything until you get to your final answer. So you can just leave it like this, but then keep this in mind that this is roughly what the numbers are around if you find it difficult to understand the first number. Okay, now that we've got this, we can get the surface area. So the way we figure out surface area is just we gotta do 0.053 times 6, because it's a cube, and then we're going to times it by 0.053 again because they have 6 faces. Yeah, I'm not the best at drawing, but you can see you're just basically getting the area of one surface and then timesing it by 6 sides of the cube. And when we do that, we get 0.016854, or which is roughly 0.017, but then again, I would recommend you leave it unrounded for now. Okay, now to get the surface area to volume ratio, because it wants us in the form n to 1, right? So the way we get n to 1 is we have to divide the two numbers, and the way we do it is we divide this this number, the bigger one, by the smaller number. So we're getting 0.016854. We're just dividing that by 0.000148 877. And when you do that, you get the number 113.221, or yeah, and depending on your rounding. So this is the part where depending on your rounding, you get that, or you get 113.33. Either way, you're going to have to round it depending on significant figures. So you have to get an answer of 113.2 or 113.3. Okay, and that is going to be what your answer is. Both work depending on how you rounded it. Generally, I recommend you don't round it until the end, just for safety, because sometimes it could lead to errors. But yeah, and that gives us our answer of 113.2 or 113.3. Only write one in your actual paper depending on how you did it, but since I'm just showing you all the answers it could be, that's why I wrote both. Okay, pretty easy question. Next one. Animal cells, unlike plant cells, do not have a cell wall. Explain how this difference affects red blood cells when placed in distilled water. Okay, so the key thing they want us to look at here is the fact that it's distilled water. And the reason that's important is because distilled water, I'll just draw a glass here. Distilled water, right? Let's say this is distilled water. Inside distilled water, there is supposed to be no solutes. There are no little solutes in there. It's just water, 100% water. And this very low solute concentration, or zero solute concentration, means that when we put something like a red blood cell inside, osmosis is going to occur. And that's what it wants us to talk about. So it wants us to talk about how water, when you place the red blood cell inside this distilled water, I'm placing it here, the water from the outside, this distilled water, will enter the cell. And we want to mention the process due to osmosis, right? That's why it's entering the cell. Because inside the cell, we have a lower water potential, right? The water is moving from high to low water potential. And that's what's happening. So let's make a reference to that. It's essentially moving from a low to high solute concentration or a high to low water potential. Either way, you can say it. I'll just say it like this. So it's moving from a low to high solute concentration. So the solute concentration is very low here, low, and this one is high, comparatively. So the water is going to move into the red blood cell, right? Because that's how it moves via osmosis. And it's asking how it's going to affect red blood cells. So let's talk about the consequence of this happening as well. Because how many marks is it? 3 marks, yeah. So we need to make more points. So what this happens is the red blood cell will swell, right? Because it's being filled with water. It's going to swell, it's going to become bigger, full, and what happens is, due to it not having a cell wall, when it swells up, it expands. The cell wall usually provides structure. It prevents it from expanding when it fills up in water, but since the red blood cells don't have a cell wall and only a cell membrane, it starts to expand, and when it expands, you know what's going to happen. Just like a balloon, it will burst, right? So this RBC, the red blood cell, will burst, right? And we can say, as a result of not having a cell wall. So yeah, pretty easy. That's all the marks we can get for the question. And yeah, well answered. Okay, explain how this difference affects red blood cells when placed in a concentrated salt solution. Now, this should be very easy to you to answer, especially considering we just did the last one. So now it mentions concentrated salt solution. Now, if you think about it, that's literally the opposite of distilled water, because distilled water is very low solute concentration, concentrated salt solution, that's high solute concentration. So we know that water entered the cell in the first case, in the previous question, so what's going to happen to water this time? Water is going to leave the cell. Water will leave the cell, the red blood cell, via osmosis. And let's talk about the water potential briefly, talk about it. So we just need to say the opposite of what we said here. So we're saying it's moving from a high water potential inside the cell to a low one in the salt solution. And similarly, we can say that it's moving from a low solute concentration inside the cell to a high solute concentration inside the solution. Because if you think about it, this salt solution we have here, this salt solution has a very high concentration of solutes, very high, it has a lot of solutes. So water is going to leave our red blood cell, it'll leave the red blood cell, and it'll go to the solution, because when it moves through the semi-permeable membrane by osmosis, what happens is it moves from a high water potential to a low water potential, or a low solute concentration to a high solute concentration. And that's all we have to say. You can make a brief mention maybe of how that will leave the water will leave the cell and the cell will die, but that's really unnecessary because it's only 2 marks, and we've gotten the 2 marks here. Okay, the diagram shows a human reflex response to touching a hot object. Some of the structures in the reflex arc are labeled. Give the name of this reflex response. Now, this is an interesting question because you might not have an immediate answer. There's a couple things that might pop in your head, and I suggest you just say them because they'll usually be right. So when I saw this question, the first thing that I thought of was involuntary. That's one of the answers. Involuntary. This is an involuntary response. You're not actively thinking to move your hand away, but your body does it to protect you. Now, some other things you can say is it's a withdrawal response because you're withdrawing your hand from the hot object in order to protect yourself. It's also a somatic response. It could be a simple response, an automatic or unconscious response. So many ways to say it, but I just went with involuntary. Okay, now draw an arrow to show the direction of the nerve impulse at point X. Now, this is just very simple. We see that this is the spinal cord and this is the muscle, right? So we're going to have to move away from the spinal cord and towards the muscle. So their nerve impulse is just going to move towards here, towards the muscle, the direction of the muscle. Okay. Which structure detects the stimulus? The stimulus is detected by structure E. It's going to be whatever is the thing that's in contact with the stimulus. And you can see that the nerves in the finger are going to be detecting it. So it's E. Okay. Which structure is the motor neuron? Motor neuron, let's see. The motor neuron looks to be I. And the reason is the motor neuron is what causes the muscle to contract, causes the muscle to move the hand away. And we can see that that's the path that it takes here. The motor neuron is the thing that's transmitting the message. So I is the thing that's transmitting the message. So it's I. So it's C. Okay. A neuron is 1.1 meter in length. The speed of the nerve impulse is 120 meters per second. Calculate the time for the impulse to pass across the neuron. And okay, well, this is very easy. We just got to take our length and divide it by the distance to get time. We have, divided by the speed, actually. So we have the length dividing it by the speed, because if you remember, yeah. So if we do distance over speed, it gives us time, and we want time. This triangle is pretty helpful, so just remember it. So 1.1, and we're just going to make sure our units are the same. So this is 1.1 meters, this is 120 meters per second. So we have the same standard of units. So we just do this, we get an answer of 0.0092. And it wants us to give the answer in standard form. Don't forget that because we want the response. And that gives us 9.2 times 10 to the minus 3. Okay, pretty simple. Describe how the impulse passes from neuron to neuron in the reflex arc. Okay, so we know that between neurons we have something called a synapse or a synaptic cleft. That's essentially what happens. So the impulses pass through a synapse. Okay. Now, what else can we say? Well, we can say that the neurotransmitter is what actually transfers the message between the synapses. It's a chemical signal. Travels across synapse. And this is what gets the message from one neuron to another. It continues the impulse. Sorry for my handwriting. There we go. What else can we say? Well, how does the neurotransmitter get across the synapse? Well, it's through a specific process which you should know, which is diffusion. Via diffusion. So it's moving across the synaptic cleft, the synaptic junction. It's moving across from a high to low concentration via diffusion. And there we go. Very simple. We got more than 2 marks, but we only have 2 marks available. So there we go. Pain has a survival function. If a person damages their ankle, they sense pain. Explain how sensing pain benefits an organism. It's a good question. So this one just requires you to think. You just think, why would being able to sense pain be helpful? Well, a simple thing is it prevents you from further damaging yourself. Let's say you break your arm, right? If you didn't know your arm was broken and you continued using that arm, your arm would get broken further. But since there's so much pain associated with that, you can't use your arm anymore, preventing you from injuring it further. And just by that logic, we can get 1 mark easily by saying it prevents further damages. Okay? Now, for mark 2, what else can we say? Well, let's look at what they said in the question. If they damage their ankle, they sense pain, right? Let's just apply it directly to that. If you damage your ankle, how does pain help you preserve your ankle, prevent damage to your ankle? Well, pain stops movement, right? If your ankle's in pain, you don't want to run anymore. You physically can't run anymore, which keeps you from damaging your ankle any further, okay? Now, that's already 2 marks, but what else can we say? Because we always want to know everything we can to get marks. Well, along these lines of preventing further damages, it allows you to rest. It allows you to experience recovery, because when we have pain, we don't want to go out and further damage whatever we hurt, which gives us the opportunity for that thing to heal and for it to repair. And another thing that you might not have thought about is that pain itself forces us to avoid painful situations. Because we know what pain is, we tend to stay away from situations which would cause us harm, which would cause us pain. Let's say if you got your hand burned on a hot stove or a hot pan, next time you would be more careful around pans. And it's essentially pain teaching us, well, some things are dangerous to touch and it could harm you. So pain is what's teaching us that something can harm us and preventing us from making the same mistake again and again. That one's a bit difficult to think about during an exam, but if you get it, that's very good. For the rest of these, they're very easy, so you should get 2 marks pretty easily on that. Okay, some medicines are used to reduce pain. These medicines work by preventing communication between the injured ankle and the brain. Explain which components of the nervous system may be affected by these medicines. Good question, and it's an application question. We have to apply our previous knowledge to the specific case of pain signals and our nervous system. Okay. So the first thing that you probably think about is the way that these medicines would work is by preventing the detection of pain, right? So that the detection of pain occurs when we are looking at a stimulus. Like in here, our stimulus is this hot object and we detect it by these nerves, right? So this medicine could prevent detection of stimulus, right? Because if there's no stimulus at the start, then the whole pain impulse would never occur, okay? And this prevention of the detection of stimulus would happen by blocking or tampering or hindering the receptor, right? Our receptor is not able to detect the stimulus, and that prevents this pain sequence from occurring, okay? Now, how else could this occur? Let's think further down the line. So if this impulse goes through and it's going through our nervous system, how can we prevent it? Well, we can prevent the transmission of the impulse. Now, how can we prevent the transmission of the impulse? So let's just write that first. Prevent the transmission of the impulse. Okay? So we've just written three things that are pretty good answers, right? You have to think a decent amount for them. But from now, we can just turn our brain off and list things that we know, because it's asking which components of the nervous system may be affected by these medicines. We've already said the receptor, that's one component, and we've explained why the receptor is. Now we've explained that we could prevent the transmission of the impulse, but now we can just list down everything we know that could prevent the transmission of the impulse. So what are the different neurons that we know, or different things in the nervous system that could affect this? Well, we know there's a sensory neuron. We know we have a relay neuron. We know we have a synapse, and that's already done. We already have enough marks, more than enough marks. We only need 4, and we've listed 3 things here, and we have 3 things here. So yeah, pretty simple. We just want to get to a point where we can start listing the different components, but make sure we give context as well. Context to how these components could be affected by these medicines. There we go. Okay, red blood cells and white blood cells are 2 of the components found in human blood. State 2 other components of human blood. This should be snapped right in your head. First thing first, plasma. Should know plasma's there. And then also platelets, the 2 P's. If you're struggling to remember the 2 other components, remember the 2 P's, plasma and platelets. Okay, B. Some white blood cells destroy pathogens. This diagram shows 4 stages in this process. Describe what's happening in each stage of this process. Well, you should know what type of a white blood cell we're talking about here, what type of leukocyte we're talking about here, because it is a phagocyte. We know that because of the way that it is ingesting this bacterium. So it is a phagocyte. Free mark there. Just list what you know. Phagocyte. Okay. And it's occurring through phagocytosis. You might not know that, you might know that, but it's essentially this form of endocytosis, the way it's quote-unquote eating the bacterium. The process is called phagocytosis, the way it digests the bacterium. But we're not going to mention that. We're going to mention a few simpler ones. So what happens? So we can see in stage 1, it's engulfing the bacterium. Okay, pretty simple, pretty like just a pure observation. And what happens, we can see in stage 2, is the bacterium comes inside of the phagocyte. Okay, there we go. Now, that's for stage 1 and 2. Let me just 1 and 2. Let's move on to stage 3. Now, what's happening in stage 3? Let's take a look. Well, we can see stage 3, something's happening to our bacterium. What's happening is the vesicles inside of the phagocyte are fusing with the bacterium. So let's see. The vesicles are fusing with bacterium. And what this leads to is, whatever it's 7, sorry. 5. The enzymes digest the bacterium. There we go. Sorry. Oh, bacterium. Okay, and this is number 3. Okay. Now, let's look at what's happening at stage 4. So at stage 4, it looks like we have exocytosis being released, and what's actually happening is the products of this digestion, the bacterium is being digested, and it's being released from our white blood cell. So products of the digestion are released. Very simple. We're just basically stating what we see, and we get easy marks. We stated way more than we need for 4 marks, but it's all good. We're doing a good job on this paper so far. So let's keep on going. See, the picture shows a human red blood cell. The actual diameter of this human blood cell is 8.1 micrometers. Describe the magnification, determine the magnification of the picture, and it's given us the conversion here. Okay, so due to the fact that I'm doing this on an iPad, I'm not sure how this will work in terms of the scale, if I'll get the right measurement, but I will measure with my ruler anyways to show you how you would do it. So we're just going to look for the diameter. So you don't want to measure it from up here. Remember, the diameter, we're looking for the longest point, the middle, right? So over here, somewhere here, and we're looking at something around 5.7. Okay, so let's get rid of our ruler, and let's write 5.7. 5.7 cm is what we have, centimeters. Okay. Now, first things first, we can't be using centimeters for our calculations because everything else is in micrometers. So what do? We do 5.7 times 10,000. Okay, because we know that 1 millimeter is 1,000 micrometers, and from that we can get the conversion that 5.7 centimeters, we times it by 10,000 to get the number of micrometers. So now we have 57,000, okay, 57,000, and we want to do our magnification calculation, and the way we do it is to get our measured length in micrometers, our measured length, and we're going to divide it by the actual diameter. So we're doing 57,000 divided by 8.1, right? That's the calculation we need to do to get magnification. And what we get is 7,037. And that is our magnification. This is also a ranged answer, so depending on your measurement, you might get different ones. Have no worries. Don't worry about getting a bit off. There is a range to the answers, but if you measure completely wrong, you won't get it right. So make sure you're taking care of your measurements. Yeah, if you wanted to know, the range is this: 6, 7, 90, 2, 7, 2, 8, 4. That's the range. So if you got an answer for magnification or anything like that, you'll be fine. There we go.